Photosynthesis consists of two phases, light-dependent and the Calvin cycle. There's many names to the Calvin cycle, but for the sake of my own sanity and everyone else's, it'll stay the Calvin cycle here. The light reaction's main purpose is to create energy to fuel the Calvin cycle. It takes place in the pancakes of the chloroplasts, otherwise known as the thylakoids. Looking at the formula for photosynthesis, we can see that we need carbon dioxide, water, and sunlight in order to create the reactants. Here we're only going to be using water and sunlight. When H2O fills a plant cell, it goes into a water splitting site inside of photosystem 2. The oxygen is released back into the atmosphere through the plant stomata, while the hydrogen now forms a proton gradient in the thylakoid membrane. The electron from the splitting of water goes into photosystem 2, where light strikes it and excites the electron to go into the electron transport chain 1, where it is then transported into photosystem 1. After this, light strikes photosystem 1 to excite the electron again, where it then goes into electron transport carrier 2 in order to be dropped off into the protein carrier NADP+, in order to create NADPH. Meanwhile, the hydrogen in the gradient is now flowing up through ATP synthase, making it spin and combine a phosphate group with ATP in order to form ATP. Now we have two energy Calvin cycle. The second phase takes place in the stroma of a plant cell's chloroplast, where it ultimately creates glucose. It uses the energy made by the first stage. If a thylakoid is like a pancake, the stroma is like syrup around it. The Calvin cycle consists of three steps: carbon fixation, G3P synthesis, and RUBP regeneration. Now we're going to be focusing on the reactant of carbon dioxide. In carbon fixation, this carbon is bonded together with a 5-carbon sugar called RUBP using an enzyme called Rurisco, where it then forms 3-PGA. Next, in G3P synthesis, 3-PGA is then synthesized using the energy from the electron carrier NADPH and ATP in order to create G3P. These energy sources come from the first step in photosynthesis. We needed 6 of each energy sources in order to create G3P 6 times, but we only need 2 3-GP in order to create glucose. So from the 6 created, 1 is taken and the other 5 continue on through the cycle. In RUBP regeneration, these 5 G3P now are converted back into the starting molecule, which is RUBP. This process takes up more ATP. This is what the...
As you can see, this is the glycolysis pathway. It starts as glucose and ends with pyruvate. During glycolysis, glucose ultimately breaks down into pyruvate and energy. Glycolysis is found in the cytoplasm. Glycolysis is a central metabolic pathway that is used by all cells for the oxidation of glucose to generate energy in the form of ATP. Glucose, 2 molecules of ATP are hydrolyzed to provide energy to drive the early steps, before molecules of ATP are produced in the later steps. The substrates of glycolysis are glucose and ATP. The enzymes of glycolysis are of the mitochondria into the intermembrane space. The second step of cellular respiration is pyruvate oxidation. It occurs after sugar is converted to pyruvate in glycolysis. The picture shows the flow of energy that occurs during this process. Pyruvate oxidation takes place in the mitochondria. There are 3 main steps in the pyruvate oxidation. 1. Carbon dioxide is released from pyruvate. 2. The remaining portion of pyruvate is oxidized to form NADH from NAD+. 3. The oxidized acetyl group binds with coenzyme A to make oxidizes acetyl CoA into carbon dioxide and provides a number of high-energy intermediates. The inputs substrates of pyruvate oxidation are pyruvate, NAD+, and coenzyme A. The products are carbon dioxide, NADH, and acetyl CoA. In a multi-step reaction, catalyzed
cycle is the third step of cellular respiration, and it's inside the mitochondria, and it happens because it needs to create FADH2, ATP, and ADH for the ETC, and the steps are acetyl-CoA combines with oxaloacetate, then citrate is oxidized, and then there's the regeneration of oxacellulose, and then the substrates, well, the primary substrate is acetyl-CoA, which combines with oxaloacetate, and the enzymes are all the gold-plated words on the diagram. And the summary of products are that 2 FADH2 goes to the ETC, 2 ATP, and 6 NADH go to the ETC, and 4 CO2 are created. The electron carriers are FAD. Phosphorylation is substrate level phosphorylation, and it works by converting the succinyl CoA to succinate. Then the energy is released from this reaction directly to phosphorylate a molecule of GTP, then converted into ATP. The fourth step is the electron transport chain, and is located inside the mitochondria, and it happens because to transfer the energy from the electron carriers to create ATP and water, and the energy came from the Krebs cycle. The steps are the electron carriers are oxidized, and then final electron accept their oxygen, which creates water, And then chiasmosis happens where ATP synthase makes ATP. The products are ATP molecules and water molecules. ATP synthase works by using an electromagnetic gradient of protons to synthesize ATP from ADP, and the electron carriers are NADH and FADH2, and they carry the electrons. How do proton pumps work? The electrons are going through to make their way the ETC through proton complexes in a series of redox reactions. While this happens, the proteins pump H plus into an intermembrane space, which creates an hydrogen ion gradient. The kind of phosphorylation is oxidative phosphorylation, and it happens by ions are going to diffuse across the membrane from high to low concentration. This process is called chemiosmosis. The diffusion of ions is going to power the phosphorylation of ADP into ATP.